So now I would like to introduce Dr. Harsha Gunawardena. He's a senior scientist in mass spectrometry at Janssen Pharmaceutical Companies of Johnson & Johnson. And today he's going to be talking to us about enabling technologies for mass spectrometric characterization of biologics. So welcome, thank you.
Okay, good morning and welcome everyone. First of all, I'd like to thank CHI for inviting me here to talk, especially Ken Simmons. It's really exciting to be here because we don't see too many of these conferences. So I'm really excited to showcase some of the work that we've been doing, new platform methods, as well as some of the newer innovations that are happening in our lab. So to give an outline to my talk, first I will give a brief introduction to large molecule analytical development, which we call LMAD. Next, I will be describing enabling MS technologies for characterization, which is the bulk of my talk, and conclude my talk with our findings and future directions. So what is large molecule analytical development? I'm sure everybody here knows what this is, but I'll just go through it really fast. So this whole drug discovery and development starts with pre-portfolio research, where 100 to 1,000 molecules are actually picked from a given target. It then goes through lead generation and lead selection, where it gets narrowed down to a couple of dozen molecules, and then finally ends up for about less than a dozen molecules per target, per program, in our group, which is the Cell and Developability Sciences, or CDS. So in CDS, we bridge discovery and preclinical development and delivers the manufacturing cell line for lead candidates, which goes on to phase 1 clinical development. So you can see on the top left-hand corner, we are working with different types of molecules, vaccines, antibody-drug conjugates, various alternative scaffolds, but more recently, we've been focusing mostly on these multi-specific antibodies, which have been quite popular in immuno-oncology. So once it comes to our department, the analytical support that goes for cell line development is as follows. From transfection to the selection of the cell line usually takes about 4.5 months. It's pretty aggressive, and we heard aggressive timelines from CMC, but for multi-specific, this is pretty fast, and we are using fit-for-purpose analytical methods through each stage of this process. And what's shown on the top right-hand table is the typical assays that we run in analytical. So these are pretty much what everybody pretty much does in analytical development. But in addition to the final comprehensive characterization of the lead cell lines that happens in analytical, we also have developed high-throughput mass spectrometry methods for screening 96D first stage colonies. And I'll be describing that shortly. So you can see there are multiple programs that we're working on simultaneously, about 6 to about 10 different targets. And you can see that when we do all this in such a short time, we have bottlenecks. And we hope to debottleneck these processes by looking at various ways to speed up or accelerate analytical characterization on our end, because we don't want to be the slow step in this process. So today I'll be talking to you about some of these enabling technologies, some new technologies which are non-platform to characterize molecules, and these are pretty new, and probably most of you in the audience haven't seen this. So the first one is the automated data analysis pipeline, and these are the topics that I'll be going through today. And the data analysis pipeline that we have built in collaboration with Protein Matrix, which is a software company that we've been working for the past 5 years or so to completely automate our data analysis end of the pipeline. So we believe that the end-to-end process should start from sample preparation all the way to reporting and data aggregation. Currently, the sample prep is semi-automated, but we are hoping to completely automate this process And we are currently working with a lot of automation companies to integrate liquid handling with the mass spectrometry, where robotics will actually deliver the samples into the mass spec automatically. It's currently work in progress, but once the data is generated in-house or through the CROs that we're partnering with, it's automatically swept to the informatics pipeline that we built with Protein Matrix, which is BIOS, and it does both peptide mapping and intact mass analysis with no human intervention, and it reports this results jointly, and then aggregates this data in their own system, as well as gene data biologics, which we are using for data aggregation. So all this happens in the Amazon workspace, where we can store, share data within the members of the group, as well as across the enterprise. I'll show you a few examples of this pipeline where we've looked at different modifications, and in this case, we're specifically looking at sequence variants. One advantage of this pipeline is it can have this sequence variant analysis decision tree, which is based on filter logic that removes possible false positive hits and clearly narrows down to the true positive hit for a sequence variant quite accurately. So what's shown on the right-hand side is one such sequence variant that we identified, which was a phenylalanine to a tyrosine isomer. So these exist in isomeric forms, and this is a common misincorporation that we observe when looking for sequence variants. And this particular sequence variant exists in 3 different forms, and we were able to pick those forms through this XIC. It's completely automated. Another advantage of using this pipeline is can do something known as bottom-up reconstruction. So we have the peptide map data with all the post-translational modifications, which can be mapped back into the intact mass profile. And here you're seeing in the dark blue is what's been mapped in terms of its abundance onto the intact mass. We can also see there are some peaks that were not identified, and those were the sialylated glycoforms that did not get detected in the intact mass analysis. But conversely, we also see a C-terminal lysine that was not detected in the intact mass, but was detected by peptide mapping. The spectrum to the right-hand side is how we are using intact mass analysis for unknown or unexpected modifications. And here we did find an unexpected modification that weighs about 340. So it's labeled here, but at the time we didn't know what it was. So we then did peptide map analysis using wildcard searches on the Bionic search engine, and we were able to detect a spectrum that had neutral losses. And this is, again, fully automated. And based on this information, we can do another database search to identify the modification, which was 2 sugar units attached to a lysine that's that is oxidized, which is known as hydroxylysine galactosyl glucose. Then we went and did further analysis on the same peptide map and got the host cell protein that was responsible for this modification. So overall, this is a comprehensive workflow that looks at the same data, same samples from intact mass, peptide map analysis to give a complete view of the modifications and enzymes or HCPs that are responsible for their modification. This is another new development. I would say it's not new. The first 2 is electron-based methods, which is called electron transfer dissociation, that has been around for a while, that we use quite often to characterize certain types of PTM, such as disulfide mapping, as well as glycosylation. But more recently, there has been emerging electron-based dissociation methods known as electron capture and electron-activated dissociation, that can also look at modifications that are isomers that exist, which cannot be differentiated by any other type of dissociation. So using diagnostic ions, using electron-activated dissociation, we can differentiate isoaspartate from aspartate, as well as leucine from isoleucine. So we are hoping to add these types of new dissociation methods to our toolbox to comprehensively characterize. This brings me to the next topic, which is rapid digestion. So this was a collaboration with an academic group at Stanford, where we were able to perform these microdroplet reactions in real time. So in the first scheme, we can introduce the immunoglobulin and an enzyme. In this case, it was IDES for subunit analysis, and we can perform these reactions in merely 4 seconds on the droplet. We can also introduce them separately using 2 sprayers, and the droplet fusion center facilitates the reaction to completion, and this takes about 250 microseconds. So we perform 2 types of reactions, the ICE subunit analysis as well as PNGS FD-glycosylation. Recently published this result, and what you're seeing on the right-hand side is how we introduce these samples using a syringe pump. We can mix them in a micro T, collect the droplets, reconstitute them in acidic solution, and introduce into the mass spectrometer. This takes 4 seconds at ambient temperatures. The same reaction when performed in bulk for 5 minutes is shown on the right-hand side of this scheme, and the spectra corresponding to each of these reactions is shown at the bottom. So you can clearly see that there was no digestion in the bulk solution after 5 minutes, whereas you can see complete digestion at 4 seconds at ambient temperatures for microdroplets. So typically, ice digestion in bulk takes about 30 minutes to about an hour. So given these time scales, we can say that microdroplets is about a 450x faster than the typical digestion. So now we've combined these reactions sequentially, where we can deglycosylate the molecule and then do ice digestion in combination with a reduction step to get these subunits. And what's shown at the bottom is these subunits that are generated using this sequential reaction. Now, we don't need to deglycosylate. We can just do subunit analysis in this case, where you can see the FC that has the different glycoforms. And then we can also do the same thing using the 2 sprayers. The efficiency is not as good, but we can still get all the 3 subunits in this analysis. The biggest drawback to both these methods that have been published is that we're introducing the sample using syringe pumps. And this is not quite amenable to high throughput analysis in a pharmaceutical setting. So this is all new work which has not been published, is that we've used this methodology and introduced the samples using an Agilent autosampler, where we are using autosampler injector programming to pick the enzyme and pick the IgG, mix them in the loop, and introduce into the mass spectrometer. And what we've done is modify the electrospray source to perform electrosonic spray ionization, which will produce the microdroplets. And since there is no quenching step, what we are doing is introducing the molecule and its fragments directly into the mass spec under native conditions. So we need to extend the mass to about 30,000, which we can do on the Agilent 6545 XT. So this is the data that we get from this, almost near complete digestion of the antibody within a cycle time of 1 minute, and that's the time it takes to do the injection. But the microdroplet reactions takes instantaneously in less than 4 seconds. In addition to these complete digestion profiles we can also get partial digestion, where we are only cleaving 1/2 of the heavy chain, and we can get IgG intermediates. Rapid injection is another topic I'd like to discuss, and how we are using that to accelerate cell line development. So some of the multi-specifics analysis is quite challenging, and in the 96 deep well colony screenings, we do this in order to distinguish the amount of impurities that are present due to homodimer formation of these antibodies. So we are trying to produce a heterodimer, but in the process we are also generating homodimers due to a mispairing of chains. So in order to accelerate the analysis, we moved away from LC-MS to rapid-fire MS for high throughput analysis, where cycle times are in the orders of 8 to 10 seconds. We can even go faster, up to 2 seconds per sample. So this is the workflow for this high throughput analysis, where we are getting the 96 deep well colony products from the 96 deep well colonies, running them through assay map to do protein purification, and doing rapid fire analysis. And our data analysis pipeline rapidly processes the data and rank orders the clones based on not just the different attributes that we are measuring, such as mispairing of chains, but also other product quality attributes such as glycosylation, as well as the titer information that we get from our CLD colleagues. So here's a good example where we examined this, where 13% of the colonies that were high titer did not go through to the final amber bioreactor stage due to product quality issues. This was due to the formation of, or detection of, half-mers and homodimers that we were able to detect through this high throughput screening method. So now I'll pivot a little bit to how we can use high throughput mass spectrometry, not just to look at chain mispairing, but also to look at product quality. In this case, we are looking at glycoforms, and we can do very rapidly in 20 minutes for a 96-well plate. So we can run it through a pipeline, get the deconvoluted mass spectrum, and look at the glycoforms. This is critical because most of the multi-specific molecules also have high levels of mannose 5, which we monitor through this process. Here I'm showing how 10 plates, 10 96-well plates, 960 spots, can be analyzed in roughly about 3.5 hours with a failure rate of 0.4% and a precision of less than 1%. So this reduces the touch time and gives us the opportunity to deliver these colonies very fast, very rapidly to our stakeholders. So if you combine rapid fire analysis with rapid automation data analysis, we can actually complete the analysis of 96 well plates in roughly about 50 minutes. So if you look at the LCMS analysis, it takes about 8 hours to do the same 96 well plates, given the run times of about 15 minutes, which is our typical runtime for reduced mass analysis. But if you know the first two workflows, A and B, has lag times because the data analysis is not completely automated. So what we are expecting by doing this rapid fire with automation is to have a immediate, handoff of the baton, if you look at this cartoon, the sprinters, so that we can come to the finish line earlier. So the third, or the C, is about 10 times faster than the LCMS run, and we can provide faster decisions to our stakeholders running through this scheme. Native mass spectrometry in combination of ion mobility is also important in deep characterization and structural analysis of our antibodies. And we are beginning to learn that this is very important in the analysis of multi-specific antibodies. So what you're seeing is native mass spectra obtained with extended mass analysis of these multi-specific maps, which has a SCFE domain and a FAB domain. And we believe that the SCFE domains are sticky, and it's known widely that they cause aggregation. And here we are seeing both dimer and trimers of this multi-specific map. So in order to further understand what motifs are causing this aggregation, we can do selective digestion. In this case, we are using an enzyme that cleaves above the hinge disulfides to get the heavy chain 1 and the FC and a Fab prime. And then we do not have the consensus on the HC1, but the digestion efficiency is rather poor, but we do get minor products of the CFE alone. So this is what we got when we ran the CCMS, which is shown here, is the fluorescence spectrum, where we saw the dimer of the heavy chain 1 and the FC in this peak. And if you look at the leading and the trailing edges of this and run them through an ion mobility technique known as cyclic IMS, you can actually get the structures. You can form a more compact structure on the leading edge, as well as a more elongated structure along with the compact form at the trailing edge of this peak, of this dimer. So these are substructures that we don't necessarily get in solution. Now, if you noted that the CFE can also form a complex with the heavy chain 1 and the FC that's free floating in solution, and that's what we're seeing here. But if you look at the SEC profile, we see this type of dimer as two distinct peaks. So peak 1 and 2 is the XIC profile right underneath the fluorescence spectrum. And if you look at the ion mobility mobilograms, you can see there is a peak 1 is composed of both a compact structure and an elongated structure. We believe that this is a gas phase artifact where the elongated structure is interconverting to a more compact structure, whereas the more predominant compact structure is the more predominant form in peak 2. So this is the kind of information that you can get when combining native mass spec with ion mobility. In addition to this, we are also looking at very high resolution ion mobility devices such as structures for lossless ion manipulation, and I can see many others who are in the audience right now who are going to talk about this in the talks in this session, where we can actually do very high resolution mass spectrometry, look at substructures within a particular domain. In this case, a Fab domain, we were able to see differences, an elongated version versus a more compact version. We've also collaborated with the Pacific Northwest National Lab, with Dick Smith's group, where we published last year, where we use this technology to look at antibody-drug conjugation. And we can obtain DAR values very accurately with very short run times of about 1 minute to obtain the DAR value. And this is a very high throughput way to determine DAR compared to SCCMS, which takes close to about a half-hour run to do this. Multiplexing. So this is my final topic, is to describe multiplexing, and very excited to share this with you, where this has not been typically done in pharma settings. It's typically done in the proteomics community, but we've developed a workflow not just to use multiplexing to quantify, but also to do two other things, namely normalization as well as identification of the conjugated peptides. The workflow starts with a sample and a mock, and we are first doing a reaction where we can actually do iodo-TMT labeling. So it labels the cysteines, and this is particularly useful for looking at cysteine drug conjugates. And then we do a trypsin digestion and do a second labeling of TMT, where it labels the N-termini or the lysine. And then we mix and run LCMS, and what we see as a spectrum is the reporters, which gives us the occupancy. So we can use occupancy estimation both at the protein level as well as at the peptide level. Now, you might not be apparent to you why we need to use two different types of labeling straight away, but what we do is we are using these reporters as barcodes, which are shown here. Is in the first case, there's 4 reporters, and that's characteristic of a peptide that can be used to quantify, estimate occupancy. In the second case, if there is a barcode where there's 2 reporters, it can be used to normalize. And in the last case, if there is 1 reporter after doing MSMS, that's characteristic of a peptide that has a drug attached to it. So this is for 1 sample, but we can multiplex up to 4 different samples. And here is a scheme where you can see the math is simple, is that the number of barcodes for occupancy is a 2n plus 2. For normalization, it's a n plus 1. And for identification is the number of barcodes is the number of samples, which is n. So the identification is done using a triggered MSMS approach, where you can do a repeat mass spec fragmentation on the peptides that only have the drug in the case of detection. Here are some examples of how this works. Here's what we get with occupancy. We can get the occupancy estimate using iodo-TMT, which is in orange, or the TMT, which is in blue, using these equations. And in this case, at cysteine 193, the antibody, the drug was conjugated at 50%, if you look at the intensity differences. Likewise, we can do this across the molecule for different sites. So it's a specific quantitation of all the cysteine conjugates. And in the second and third case, there's no intensity difference, and therefore there is no conjugation. And this is how we use the non-cysteine peptides now to do normalization. So you will only get a barcode of 2, and in this case, it was mixed properly. So this is essential in this workflow because there could be digestion differences or mixing errors. And this facilitates us to normalize for any errors that we introduce during sample prep, all automated. And then I'll finally describe this, the third element of this workflow, which is detection. We are doing report ion triggering on the barcodes. And in this case, it's a single barcode for a single sample. In the first scan, it's a full MS1 scan. In the second scan, we are doing collision-induced or high-energy collision-induced dissociation to generate the reporter. And only upon seeing the reporter, it triggers the electron transfer dissociation spectrum. So within the elution profile of a peak, you can see that we can collect MSMS data on the peptides that contain the drug quite selectively. So all this happens based on this logic of reporters that we specify in the analysis. And this can only be done currently on a Lumos instrument, unfortunately. So these are data that I can again show. It's the HCD spectrum, and you can see the reporter at 130. It's a single reporter. And the disadvantage of this method is if you just look at HCD, you can also see the drug that's conjugated also fragments. But if you do a triggered MSMS scan with electron transfer dissociation, you can very accurately localize the drug without the drug fragmenting. So we've done site-wide quantitation where we can actually estimate occupancy for all the cysteines in this conjugated drug. And in the second volcano-like plot shows that the non-cysteine peptides can be used for normalization. So the light chain cysteine that's conjugating the drug had an occupancy that was validated also by SLIM and with Dick Smith's group, where we did estimate the occupancy using the mobilograms. So now we can multiplex up to 4, and this is a scheme to do that, and we can completely automate them using AssayMat robot to do the labeling as well as the digestion. And here's a quick result showing the use of multiplexing using TMT 10-plex reagents, which are isotopologs. And if you look at closely, the mass difference between the two reporters for 1, 2, 3, and 4 is about 6 millidaltons. So you need high-resolution mass spectrometry to do the occupancy estimate. For anything more than two samples, up to 4, you need this type of mass resolution. So with that, I would like to conclude my talk. We've demonstrated that MS technologies enable both throughput, analysis speed at various stages of biologics development. We've increased the speed several million-fold using microdroplets. Sample introduction for screening product quality assessments were done using rapid fire. Sample multiplexing was performed for accurate and fast peptide quantitation. And alternative dissociation was used to improve product characterization, as well as native ion mobility mass spectrometry was used to understand the structure of the antibodies. So we are continuously developing and refining analytical methods, informatics, to reduce the timelines and improve the operational efficiencies. We are looking at better ways to perform all these things, integrating mass spec with robotics, as well as integrating various assays in an autonomous fashion. So that's going to be the future for the next couple of years. And with that, since we are almost out of time, I would like to acknowledge all the members at Janssen, as well as a lot of our collaborators who are also in this room. I would like to answer any questions if you have. Thank you.
We have about 2 minutes for questions. Do we have a quick one for Harsha?
There's one there.
Oh, I'm sorry.
Hi, I'm Nancy Fernandez from Lonza. I'm interested in the Pico spray, and I was wondering with your microdroplets if you've ever tested any of the PPNL Pico spray devices, or are the pneumomics commercially available?
Yeah, we've thought about it. So that's a good way to generate droplets, but the smaller the droplet, there's high efficiency of the reaction, but we don't want to make it too small, because if it's too small, there's no time to do the digestion. So the residence time is also important. So one critical parameter that Dick Zare from Stanford and Graham Cooks have shown is that the distance on the sprayer to the inlet is also as critical as the size of the droplet. So we don't want to do too much of drying on the fly, so that we don't quite reduce the efficiency of the reaction. So the mechanism is still up for debate. We're also looking into that along with these academic partners, because this was the first time we did an enzymatic reaction. So Graham and Dick, they are focused mainly on examining organic reactions and accelerating, improving the reaction efficiencies of difficult organic synthetic routes. Never been done on a antibody or on an enzymatic kind of reaction. So this is the first. And we still are trying to figure out some of the parameters to improve the efficiencies. Yeah.